Hello, my name's Kirsten McBain and I'm a scientist on the Bioanalytics Applications team at Sartorius. And today I'm going to be presenting a poster that we took to the AACR meeting this year titled Comparative Analysis of Anti-HER2 Antibody-Drug Conjugates in Tumor Spheroid Models. It is clear that with the continually rising emergence of antibody-drug conjugates, we need robust techniques to screen their activity in vitro. And sometimes the traditional monolayer models that are used, whilst they're cost effective and scalable, they can lack features of the tumor microenvironment. So for example, complex cell-cell interactions or also interactions with the extracellular matrix. And because of this reason, tumor spheroids can provide a more translational model for ADC quantification and can lead to a more realistic representation of drug effects and a relationship to the in vivo environment. In these data, we have taken temporal measurements of ADC internalization and cytotoxicity using the IncuCyte live cell analysis system. At the assay endpoint, the spheroids were dissociated and then analysed using the IQ high throughput screening cytometry platform. And this allowed us to yield further information on spheroid health and composition. This combined workflow allows us to distinguish ADC mechanisms of action using both single and multi-spheroid models. I'm going to start off by talking about work we did to look at internalization of antibodies into single spheroids. So for this workflow, we started off by seeding cells, which in this case were HER2 positive BT474 cells from a breast cancer cell line, into ultra-low attachment plates for 72 hours, which leads to the formation of single spheroids. And we monitored this spheroid formation using the IncuCyte, by which we can take images. So in this case, we took them every 3 to 6 hours, and we could monitor that spheroid formation and growth. On day 3, we labeled antibodies. So the antibodies we were using in this case were trastuzumab, which is an anti-HER2 IgG1 antibody. There was Kadcyla, which is an ADC based on trastuzumab, and then also Enhertu, which is another ADC based on trastuzumab. And what we were labeling them with was the IncuCyte Human FabFlor pH Orange antibody labeling dye. So basically, this is a pH sensitive probe that labels the antibodies. And then when those antibodies are internalized into acidic endosomal and lysosomal pathways, they see an increase in fluorescence. So we can then quantify that increase in fluorescence over time using the IncuCyte, and then we presume that this readout correlates with an increase in antibody internalization. And so starting with the images in the middle, you can see there's both phase on fluorescence and fluorescence only images. And it's clear straight away that all of our HER2 targeting antibodies, so the trastuzumab and the two ADCs, have an increase in fluorescence when those labeled antibodies were added. Whereas when there's no antibody and the IgG control, there's clearly been no increase in fluorescence. And in the bottom right, you can see a quantification of this time course, which shows that the internalization of the two ADCs is fairly comparable at a high level, whereas there's slightly lower level internalization with the trastuzumab antibody. And this is actually an effect that we've seen now both in 2D and 3D models. And it's interesting because, of course, the internalization capacity is essential for the ADC function, although we're not entirely sure why the internalization of these antibodies may be enhanced. The next set of experiments aim to use a multispheroid model in order to quantify the cytotoxicity of these antibodies. So for this workflow, we began by coating a flat bottom plate with Matrigel. And then on top of this, we added a suspension of our cells, which in this case were Incucyte nuclite green lentivirus cells, and they were either SCOV3s from a high HER2 expressing line or MDA MB231 cells from a low HER2 expressing line. And again, we used the Incucyte to monitor the formation of the multispheroids over time. And then on day 3, we added our treatments, which in this case were the trastuzumab, the Kadcyla, or the Enhertu, or an IgG control. And what we could see already from the images is that in the IgG control wells, we've got kind of much larger spheroids compared to particularly the ADC containing wells. And if we look then at the quantification, which in this case we've used the green intensity metric, you can see that with the SCOV3s, so the SCOV3s are the ones in the images, there's clearly a difference between the kind of IgG control in which there's kind of continued increase in the green intensity, whereas with the trastuzumab and the Kadcyla and the HER2, there's been a clear reduction in the kind of intensity of those green labeled spheroids. And it was interesting to note that this was not seen with the MDA-MB231 cells, showing that we are able to measure a kind of specific effect in that only HER2 expressing cells are having growth inhibition in the presence of the anti-HER2 antibodies. So here again, we're looking at ADC cytotoxicity, this time in single spheroid models. So in the same way as we did for the internalization experiment, we were seeding cells into the ULA plates for 72 hours in order to promote single spheroid formation, and then adding our antibody treatments on day 3. And again, these spheroids were formed from BT474 cells, which we added a range of concentrations of each antibody to. And again, we took Incucyte images to monitor the spheroids over time. And in this instance, kind of not in the same vein as the multispheroid experiment, we were actually monitoring the spheroid area in order to quantify cytotoxicity. So actually, usually we would recommend using fluorescence again, but in order to keep consistency with the next figure in which we were looking at kind of mixed cell type spheroids, we've just shown the spheroid area here. But we could still definitely see some differences with the in the spheroid area in the presence of these antibodies. So if we first look at the ADCs, the Kadcyla and the Enhertu, there's clearly been a concentration-dependent reduction in the spheroid size over time in the presence of the two ADCs, as we'd expect with these HER2-positive spheroids. With the trastuzumab, it really seems to be more of an all-or-nothing effect. So at our very high top concentration of trastuzumab, we have seen considerable death of this, a considerable reduction in the area of the spheroid, indicating spheroid death. However, we don't see a huge difference at the lower concentrations, and then we don't see anything really standing out with the IgG control as we'd expect. So as I just mentioned, in our final experiments, we were wanting to have a look at some kind of mixed cell type spheroids, and this is because we were hoping to investigate the bystander effect. So Enhertu should have a bystander activity, which essentially means that once it has entered and induced cytotoxicity of HER2 expressing cells, the payload should then be released and diffuse into neighboring cells, regardless of their HER2 expression, and induce their cytotoxicity as well. So to look at this, we were generating single spheroids from a 2:3 ratio of green labeled high HER2 expressing BT474 cells and unlabeled MDA MB231 cells, which are low HER2 expressors. And We again added the antibodies after 3 days and captured images using the IncuSight every 3 hours. On day 8, the spheroids were dissociated, and then we labeled them using an cell membrane integrity dye, and this meant that we could quantify the remaining numbers of both cell types using the IQ high throughput screening platform. So starting with the IncuSight data in the top, this shows a time course for the spheroid area. So, as I mentioned, because we're looking at a mixture of green and unlabeled cell types, we didn't really want to use a fluorescence metric in this instance, but we can still see that with the spheroid area, there's been only a reduction with the Enhertu, whereas the kind of overall spheroid area seems to remain fairly constant with the IgG control, trastuzumab, and Kadcyla. And then when we look at the bar charts at the bottom, this shows the IQ data. So on the left, we have the BT474, so the high HER2 expressing cells. And kind of similarly to what we've seen previously, we've seen high level death with the 2 ADCs, and then sort of some mid-range effects with the trastuzumab. In terms of the MDA MB231s, we've seen kind of minimal effect with the IgG, the trastuzumab, and the Kadcyla, but we've seen a considerable reduction with the Enhertu. So this would indicate that perhaps the bystander effect is occurring, since only the Enhertu has been able to reduce the number of the HER2 negative cell type. To conclude, we have displayed the use of the IncuSight live cell analysis platform to quantify the effects of ADCs using 3D models, specifically looking at the readouts internalization and cytotoxicity. We've highlighted how the IncuSight can be used in a combined workflow with the IQ HTS platform to investigate and compare the mechanism of action of ADCs, for example, having the ability to reveal the bystander effect. And these techniques can be utilized to inform drug discovery and biological research workflows. Thanks very much for listening.